Hi guys, Dr. Orlando here with a vidal on mitosis. I'm going to draw out the phases and say what happens in each. So if you have your notes from class, grab those and follow along in your notes. Make sure that you got everything that you were supposed to when we were drawing it on the board. Okay, so I'm going to start with a cell in interphase. So this cell is in interphase. Notice that I drew the DNA in the nucleus not as the little line squiggles or X's. It's just kind of loosely arranged in there at the moment. It's not condensed at this point. It's not organized. All right, so during interphase, you're going to see the chromosomes appearing to look like, and the word that we use in class is chromatin. So just a loose collection of the DNA and the proteins that are present there in the nucleus. Okay, but during interphase, and specifically, we're going to be in G2 of interphase. This cell knows that it's going to be going through mitosis. So it's going to, here's its original pair of centrioles. It's going to make a new copy. It's going to make a new set. We'll put little, let's zoom in on him. He's all brand new, so I'm making him look like he's shining. Okay, so a new set of centrioles is going to be made. So the second pair of centrioles is made, and we're going to start making some parts to the spindle as well. Okay, so that's interphase. If we were able to look at the chromosomes here, they would already be replicated at this point. They've gone through synthesis. So we've got sister chromatids that are paired up in that mess there in the chromatin. Okay. All right, so now let's move on to actually mitosis. So here's our line where we're going to start mitosis. So here's our cell, and now we are going to be in the first phase of mitosis, which is called prophase. Okay, and in prophase, the nucleus is going to begin to break down, so I'm going to draw him kind of dotted, and I'm going to mark this as early prophase, or early in prophase. The nucleus is beginning to break down. Really, the nuclear membrane begins to break down. Okay. The chromosomes are going to condense. So I'm going to draw, zoom in here, I'm going to draw two sets of chromosomes here. So our chromosome number for this guy is going to be 4. So I'm going to draw a large blue and a small blue, and I'm going to draw a large red and a small red. Now think in your mind, why did I do blue and red, and why did I do bigs and smalls? Well, I did blue and red to show homologous pairs. So my big blue one, I'm going to draw on this and then I'm going to erase it. These guys here, let's get a yellow, like a highlighter. These guys here are a set of homologous chromosomes. Okay, as are those guys. That's a set of, so these are two sets of homologous chromosomes. Okay, get rid of that. Okay, so those are our sets of homologous chromosomes. Remember, when I draw them in the X shape, Those guys are the sister chromatids. Each squiggle line that is part of the X is a sister chromatid. They're identical to each other. Okay, so the nucleus is breaking down in prophase. The chromatin is condensing to make those X shapes that we see. Okay. All right, the centrioles are going to migrate to the poles. So this is, if you think about this circle like the Earth, you'd have the North Pole and the South Pole, right? And you'd have an equator. Okay. That's not how and we don't have a North and South Pole to a cell, but we use the term poles referring to the opposite ends, and the equator referring to the middle. It doesn't have to be the top and the bottom. It could be side, side, diagonal, diagonal. It all depends on how the artist draws it, or how, when you look at an actual live cell or a specimen that's been stained for you to see, it depends on where the orientation of how it was cut. So don't always get stuck on tops and bottoms. Okay, but just make sure that the centrioles are at opposite sides. They're opposite each other. And then wherever they are opposite, mentally draw a line middle through them that divides them in half, like an equator. Okay, so the centrioles are being moved to the poles, and the spindle is being assembled. Do that in a different color. I wanted that in blue. Sorry about that. The spindle's being assembled. So let's add that to our last thing there. Okay. So that's prophase. Now prophase, a lot going on in prophase. It's really hard to draw in one picture and hard to give it one name. So often you'll see prophase referred to as early and late, or prophase and prometaphase, which is how your textbook does it. So we'll go with that. Prometaphase, which is late during prophase. Okay. At this point, the nucleus is gone. And the chromosomes are attaching to the spindle. Okay. So let's draw in our centrioles at opposite ends. Our spindle, draw them at different lengths because they're not all lined up perfectly yet. There's a bit of a tug of war occurring. Okay. You can draw the spindle attaching to each chromosome on each side of the chromosome. Who am I missing here? Oop. Let's get rid of that. And we need a little red guy here. Okay, and we need a spindle fiber from that side. And just because it's bugging me, I'm cleaning this up like that. Okay. All right, so the spindle is attaching to the chromosomes, and it attaches at a specific spot on the chromosome by the centromere, and it's called the kinetochore. So the kinetochore is the place on the chromosome where spindle attaches, spindle fiber attaches. Okay, so notice that we're almost there. Imagine these spindle fibers kind of playing tug of war, tightening and pulling back and forth. And when they end up on midline, we call it metaphase. Metaphase, okay? Prophase. So metaphase, you're going to draw your centrioles. Then you're going to draw your chromosomes. And I'm just going to keep the order as I drew them there. The order of these chromosomes, how they line up on the equator, is not important. It's just important that they all end up on the equator. Okay, so I'm going to draw these guys. Draw my spindle attaching. Okay, so give some words to this. For metaphase, the homologous chromosomes line up on the equator single file. So there's a single row of them. Okay? And they line up on the equator single file, as you can see, nice and neat, straight across. So from there, we go on to anaphase. It looks like I'm running out of room on this page, so I'm going to go to the next page. Okay. And we'll do anaphase here. So previously these chromosomes were lined up on the equator. Now draw your centrioles, but draw the chromosomes being pulled apart. Sister chromatids are split. They're pulled apart. So my order that I had before, just to make sure I keep them in the same order, I had the long blue, then I had the short blue, and I had the long blue, and then I had the short red. Okay. Notice how I'm drawing them like V's. So the reason that you see artists draw them like that is because that's what it actually looks like. So imagine these chromosomes being pulled apart, and they're being dragged through cytoplasm. They're being pulled through this thick gelatinous goo, right? So as they're being pulled through, they kind of fold back. Whoop, look, I drew that guy upside down. Sorry about that. He's being pulled the other way. There we go. Okay, so they're being pulled through the cytoplasm just like that. If you were to zoom in on them, they would look like this. So there's the chromosome, you know how they kind of have those ends, and there's their kinetochore. They're being pulled like that, okay, in that direction. And so the tails are kind of dragging back through the cytoplasm. So this is anaphase. We're pulling the sister chromatids apart. So sister chromatids are split and pulled to the poles. Now, how does that happen? Remember in class I drew this guy on there to help you remember. Remember him? Little Pac-Man, waka waka waka. Okay, put him there. Put him over here. So Pac-Man, as we know, chomps things in the game, right? And what happens is that motor proteins down here in the centrioles in the centrosomes Remember, the centrosome is the centriole and all of the little proteins that are around it. Okay, so the motor proteins in the centrosome pull the kinetochore toward the poles by chomping the end of these spindle fibers, almost like they're eating it. Okay, and what does Pac-Man do? He chomps. So this is actually termed Pac-Man action. So Pac-Man action chomps those spindle fibers, shortening it and bringing the chromosomes closer to the poles. I didn't do a really good job of showing this other thing that happens in anaphase, and that is that there are microtubules that are going to start stretching this cell out. Okay, so it should be, I wish I would have drawn them a little more oblong there, but what are you going to do? So let's write that down. So microtubules, excuse me, microtubules force the poles farther apart. Making the cell an oval shape. Okay. All right. Whoop. All right, so there we are, pulling the chromosomes closer to the poles and stretching that cell out, separating sister chromatids. All right, next one then. Last phase of mitosis is telophase. So we're going to draw our cell nice and oblong because it got stretched out. During telophase, you draw your little centrioles. Okay, during telophase, the chromosomes have arrived at the poles. So let's draw them in. There's a long blue, short blue, long red, short red, long red, short red. Okay, so they've arrived at their poles and the nucleus forms around them again. Nuclear envelope forms around them. Don't include the centrioles in your nucleus. They stay outside of it. Okay. So at this point, you can see I didn't draw the spindle, so the spindle dissolves. Got the nuclear envelope, the nuclear membrane, reforms. The chromosomes are actually going to go back to, even though I drew them as the little squiggles, they are actually going to go back to their chromatin state. They're going to revert. Let's write chromosomes revert. To the non-condensed state. Chromatin. Okay. You're also going to see nucleoli. Let's get a nice purpley color for that. You're going to see the nucleoli reform. Okay. There you go. Remember, the nucleoli is the ribosome factory, so you find RNA and ribosomes in all states of production. Okay. And that's telophase. So if we stayed in telophase, we'd have a cell with two nucleus. And mitosis is done at this point because mitosis is nuclear division. We divided up the nucleus. We did a good job. So from there, we have to go into the final process that splits these, and this is called cytokinesis. So in cytokinesis, we usually draw the cell starting to look like this, kind of pinched in the middle. Draw the nucleus. At this point, our chromosomes have gone to the chromatin state. They are all relaxed at this point. Okay. So we can draw them like that. Okay. So during cytokinesis, the cytoplasm is divided. Microtubules are going to form this kind of purse string around the waist of the cell, and around the equator. The cytoplasm is divided. Microtubules form a purse string, or sometimes people refer to it as a drawstring waist around the equator. Okay, around the equator of the cell, and it forms that cleavage furrow. Cleavage refers to cut, like if you cleave something, you cut it, or a cleaver chops meat, right? So cleavage means to cut. Okay. So eventually those microtubules are going to pinch off completely, and we will have two separate cells that look identical to each other. Okay? So two separate cells, and cytokinesis is done. Now let's look at the result. I know I didn't draw the chromosomes in on these, so let's put them over here. If we were to look at how it looked during telophase, okay, you can see that we have the chromosome number is 4 in each nucleus, so that would continue on along here. Wouldn't change the chromosome number at all. So at the end, the chromosome number is still 4 in each. And if we go back to our beginning, here at prophase, we see the chromosome number was 4 as well. They happened to be duplicated chromosomes, but there was only 4 of them. Okay, so we kept the chromosome number here in our cell, which is an important point about mitosis. So mitosis is going to be the key characteristic being that we are making clones or exact copies. Not changing the chromosome number at all, going from a 2N cell to a 2N cell. Okay. All right, if you have any questions, please feel free to contact me.